Hello everyone. Welcome to the next episode of Micro Talks by Manju ma'am. As we all know, viruses are unable to reproduce independent of living cells, and hence they need to be reproduced or cultivated in a manner that is different from the prokaryotic and eukaryotic cells. The prokaryotic and eukaryotic cells, as we all know, can grow on inanimate media. However, viruses being obligate intracellular parasites, so-called, they are unable to replicate without a host cell. Moreover, viruses are host specific, so they cannot be grown on any universal cell that will support the growth of all viruses. There are different methods of viral cultivation. So let us see which are the different methods of viral cultivation. Initially, in the olden days, animal inoculation was the chosen method earlier. However, there are many limitations to this method. So this method is no longer in use. The biggest limitation of this method being the host defense mechanisms that come into play whenever a virus is inoculated in a test animal for it to grow. Secondly, there are a lot of ethical issues that are involved with using lab animals for experimentation and cultivation of viruses. Chick embryo technique is widely used. It is cheaper. The chick embryos are easy to maintain. There are no host defense mechanisms involved. The embryonated eggs are readily available, and this method is not so labor intensive. In the recent past, tissue culture has replaced chick embryo technique. There are three major types of tissue culture. You have the organ culture, explant culture, and the cell culture. What is the need for cultivating viruses? Obviously, viruses need to be cultivated for diagnosis and to understand the antiviral therapies that could be used for treatment of the viral infections. Bulk cultivation of viruses is needed for vaccine production. Hence, using embryonated eggs is a good option. Virus cultivation is required in research in order to understand the replication, the genome of the virus, the infection mechanisms, the composition of the viral components, and the possible target receptors which could be used further to design antiviral drugs and determine treatment regimes. Study of different epitopes for producing effective vaccines is also possible. So there are a lot of avenues in research for which the viruses are required in bulk. Let us talk about the chick embryo technique. This technique was first used in 1931 by Goodpasture. A chick embryo has to be 7 to 10 days old. Initially, you need to sterilize the surface of the egg. You have to perform the candling to identify the air sac. The air sac will appear as a hollow and the embryo will appear with a red dot at the opposite end of this hollow. Once the air sac has been identified, the position of the air sac has been identified, the embryonated egg can be inoculated using a syringe on different parts of the chick embryo for different viruses. Once the inoculation is done, you have to seal the opening, that the little minuscule pore that has been created with either gelatin or paraffin, and the eggs are incubated at 37 degrees for 2 to 3 days. Viral growth and replication in the egg embryo is indicated by either the death of the embryo or damage to the embryo or by the formation of typical pox or lesions on the egg membranes. There are different areas of the embryonated egg that can be used for inoculation and cultivation of the viruses. You have the chorioallantoic membrane, which is used for the cultivation of the herpes simplex virus, the pox virus, and the Rous sarcoma virus. Besides that, you have inoculation in the amnion, either on the amniotic membrane or in the amniotic fluid, where you could inoculate the influenza viruses, that is both orthomyxo and paramyxo, and the mumps virus. Egg yolk is another important site for viral inoculation, the yolk sac, on which herpes simplex virus can be inoculated. And then you have the allantoic cavity and the allantoic membrane, and on the allantoic membrane, influenza virus, mumps virus, Newcastle disease virus can be inoculated. These are just some representative examples of the viruses that I am stating here, which could be used for cultivation in the chick embryo technique. Let us look at the advantages of using an embryonated egg or the chick embryo technique for viral cultivation. There is no need for any ethical considerations or licenses from the regulating authorities. It is an ideal substrate for the viral growth and replication. There are different sites on which the viruses can be inoculated and cultivated. Obviously, this method is much more cost effective, and maintenance of the embryonated eggs is much easier and cheaper. Less labor is required. The embryonated eggs are always readily available. And as I said before, there is a wide range of sterile tissues and fluids, devoid and free from any contaminating bacteria and latent viruses, which would otherwise show up in the cultivated embryonated egg. And of course, there are no specific and non-specific defense factors that are involved in the embryonated eggs, so the viruses can grow freely. Embryonated eggs can be used for maintenance of stock cultures of viruses. What are the disadvantages? There are very few disadvantages. To cite the main ones, there are different routes of inoculation for different viruses. So that could become a challenge, because if the technician is not trained well enough, the site of inoculation could go wrong. The eggs are obviously very fragile and they can easily break, so they need to be handled with a lot of care. So that's all for now on the viral cultivation using the chick embryo inoculation. Thank you so much. Please do like, share and subscribe to my channel, bringing you more of viral cultivation in the next episode, where we will be discussing the tissue culture. Thank you so much.